Hi, my name is Ashwin and my presentation is on the role of glycoproteins in cell adhesion. So general overview is are proteins which contain oligosaccharide chains that covalently attach to the amino acid side chains. This is done through a process called glycosylation. These glycoproteins are important integral proteins that play a role in cell-to-cell interactions. They are generally found on the surface of the lipid bilayer of cell membranes. They have a hydrophilic nature due to the sugar being added to the protein, which allows for the functioning in this aqueous environment. Now, cell adhesion is the process by which cells interact and attach to neighboring cells through specialized molecules of the cell surface. They attach to the extracellular matrix using cell adhesion molecules. These cell adhesion molecules are transmembrane proteins located on the cell surface, and they are very important for cellular function. Now, for the structure and the proteins involved, glycoproteins are covalently linked of the protein with the carbohydrate. They are either O-linked, which is where the linkage is using the hydroxyl group of the amino acid serine or threonine, or they are N-linked, which is when the linkage is with the amino group of asparagine. Carbohydrates can be sugars with the amino group, or sugars with a carboxyl group. They are very hydrophilic due to the OH group being there, and that is very helpful for tertiary structure folding. Now, cell adhesion molecules are transmembrane receptors, and they have two different binding interactions. They could be homophilic, where the cell adhesion molecule binds with the cell adhesion molecule that is the same, or heterophilic, where the cell adhesion molecule on one cell binds with the cell adhesion molecule on another cell. They are also classified depending on the relation to calcium. The two that are calcium independent are integrin and the immunoglobulin family. And the two that are calcium dependent are the cadherin and selectin. Now, these glycoproteins that deal with cell adhesion are primarily located on the plasma membrane. They are very common in red blood cells. For example, one of the most common ones is glycophorin A. And the hydrophilic nature, as stated before, allows them to function in the aqueous environment. This lets them act in cell-to-cell recognition and binding of other molecules, as well as very important for the structural stability of tissues. Now, there are several diseases that are associated with these cell adhesion molecules and glycoproteins. However, the most recent topic that has been researched has been its interactions with cancer cells, specifically with how alterations in the adhesion properties of blood cells plays a very pivotal role in the development of metastasis for these cancer cells. And it's also been looked at with how many cell adhesion molecules are affected by these alterations that deal with the epithelial tissue. One of the most recent findings was dealing with colon cancer and human umbilical vein endothelial cells, or HUVECs. So what these researchers looked at was they looked at colon carcinoma cell adhesion, and they looked at its relation to the human umbilical vein endothelial cells using glycoproteins. They saw that LS147T colon adenocarcinoma cell adhesion involved both the O and the N-linked glycoproteins. They also saw that there was more glycoprotein involvement than glycolipid involvement. And as well, they saw that E-selectin endocytosis was upregulated, which may reveal a possible correlation between these two. Now, a future question would be, would glycolipids have a very similar effect on cell adhesion? Thank you.